Welcome to a journey into one of the most revolutionary frontiers in modern science. Today, we're exploring how cellular senescence, the process where cells stop dividing but don't die, drives aging and disease, and how cutting-edge CRISPR technology might allow us to rewrite the story of human aging. From the molecular mechanisms of aging to the ethical questions raised by age reversal, we'll examine how scientists are using gene editing to potentially extend not just lifespan, but healthspan, the portion of life spent in good health. At the core of aging lies cellular senescence. These are cells that have ceased to divide but remain metabolically active in our tissues. Recent research published in Nature Aging has revealed that these senescent cells accumulate over time, becoming unwelcome guests in our bodies. But they don't just sit there quietly, they secrete pro-inflammatory factors collectively known as the senescence-associated secretory phenotype, or SASP. This cocktail of inflammatory molecules creates a state of chronic inflammation that gradually degrades tissue function and accelerates aging. Think of senescent cells as retired workers who, instead of peacefully stepping aside, actively disrupt the workplace by spreading negativity and dysfunction to otherwise healthy cells around them. A groundbreaking discovery from the Buck Institute for Research on Aging shows that senescent cells don't just change their behavior, they undergo profound changes in their epigenetic makeup. Epigenetics refers to chemical modifications that affect gene expression without changing the DNA sequence itself. These modifications include patterns of histone methylation and DNA methylation that scientists can now measure as part of what's called the epigenetic clock. This clock, pioneered by Dr. Steve Horvath at UCLA, represents one of the most accurate biomarkers of aging discovered to date. The remarkable thing about the epigenetic clock is its precision. It can determine a person's biological age with stunning accuracy, often revealing that some people age faster or slower than their chronological age would suggest. And this clock doesn't tick at a fixed rate, it can be accelerated by chronic stress, poor diet, and environmental toxins, while lifestyle interventions like exercise and caloric restriction may actually slow it down, suggesting that biological age is malleable. Beyond epigenetic changes, aging involves several interconnected molecular processes. One key factor is telomere shortening. Telomeres are the protective caps at the ends of our chromosomes, similar to the plastic tips on shoelaces. Each time a cell divides, these telomeres get slightly shorter. When they reach a critical length, they trigger cellular senescence, essentially setting a limit on how many times a cell can divide. Another crucial aspect is mitochondrial dysfunction. Mitochondria, the powerhouses of our cells, become less efficient with age. They produce more harmful reactive oxygen species, essentially cellular exhaust fumes, and generate less energy. This energy deficit contributes significantly to the aging process at the cellular level. Additionally, aging involves declining proteostasis, the cellular system responsible for maintaining protein quality. As this system falters, misfolded proteins accumulate, causing cellular stress and contributing to age-related diseases like Alzheimer's and Parkinson's. These processes don't occur in isolation but form an interconnected network of molecular changes that collectively drive the aging process. Enter CRISPR-Cas9, the Nobel Prize-winning gene editing technology that's revolutionizing our approach to cellular aging. Unlike previous approaches that focused on eliminating senescent cells, known as senolytics, cutting-edge research is now using CRISPR to actually reprogram these cells. This represents a fundamental shift in strategy. Rather than simply removing the problem cells, scientists are working to reform them. A team at the Salk Institute has developed a modified CRISPR system that doesn't cut DNA but instead modifies the epigenetic markers associated with senescence. Their system, called CRISPR activation, can selectively activate genes that counteract the senescence program. When tested in mouse models, this approach showed promising results: partial restoration of tissue function in artificially aged animals. Meanwhile, at Harvard's Wyss Institute, researchers have created a CRISPR-based screening tool that identified 253 genes potentially involved in triggering or maintaining the senescent state. This comprehensive genetic map provides multiple targets for intervention, expanding our arsenal in the fight against cellular aging. The versatility of CRISPR technology extends far beyond simply editing genes. Scientists at Stanford University have developed a system called Crispr switch, which can precisely control the timing of gene editing activities. This temporal control is crucial when addressing aging, as it allows researchers to target specific developmental windows or activate rejuvenation pathways in a coordinated manner that mimics natural developmental processes. Imagine being able to orchestrate a symphony of genetic changes, each occurring at exactly the right moment to optimize cellular rejuvenation. Another promising approach involves using CRISPR to enhance the activity of our body's own repair mechanisms. Research at the Mayo Clinic has demonstrated that specific DNA repair pathways become less efficient with age, contributing to genomic instability and cellular dysfunction. By using CRISPR to upregulate these repair mechanisms, researchers have observed improved DNA maintenance and reduced markers of cellular aging in preclinical models. This approach works with the body's natural systems rather than replacing them, potentially offering a more harmonious intervention. The transition from laboratory research to clinical applications is happening at a remarkable speed. Rejuvenate Bio, a biotechnology startup co-founded by renowned Harvard geneticist George Church, has begun preliminary trials using a CRISPR-based approach to target aging in dogs. Their therapy delivers gene editing components via adeno-associated viruses, OVs, to modify specific longevity-associated genes. Dogs provide an excellent model for human aging interventions due to their similar lifestyle environments and compressed aging timeline. Meanwhile, Altos Labs, backed by significant investment including funding from Jeff Bezos, is pursuing cellular reprogramming technologies that incorporate CRISPR to reset cellular age. Their approach combines partial cellular reprogramming with targeted epigenetic modifications to rejuvenate cells without completely erasing their identity, maintaining the cell's specialized function while reversing age-related damage. The regulatory frameworks are struggling to keep pace with these innovations. The FDA has established a cellular, tissue, and gene therapies advisory committee specifically to evaluate these emerging technologies, acknowledging both their promise and potential risks. This represents a significant step in creating pathways for these therapies to reach patients safely. The commercialization landscape extends well beyond the high-profile ventures we've already discussed. Unity Biotechnology has focused on developing senolytic compounds that can be used in conjunction with CRISPR-based approaches, creating potential combination therapies that both remove damaged cells and reprogram the remaining ones. Their clinical trials for osteoarthritis and age-related macular degeneration represent some of the first anti-aging therapies to reach advanced clinical testing. The economic potential is staggering. Analysts at Morgan Stanley project that the longevity market could reach $600 billion by 2025, reflecting both the enormous potential and substantial investment in the sector. This rapid growth has attracted attention from major pharmaceutical companies, with Roche, AbbVie, and Novartis all establishing aging research divisions or partnering with anti-aging startups to develop their pipelines. We're witnessing the birth of an entirely new industry focused not on treating individual diseases but on modifying the underlying process of aging itself. This represents a fundamental shift in how we approach medicine and human health, moving from a reactive model of treating diseases as they appear to a proactive model of preventing age-related decline before it begins. The rapid advancement of age reversal technologies raises profound ethical questions that society must address. A recent survey by the Pew Research Center indicates that 63% of Americans are concerned about the societal implications of significantly extended lifespans. These concerns include resource allocation in an increasingly long-lived population, environmental impacts of population growth, and potential socioeconomic divides in access to longevity treatments. Will these technologies be available to all, or will they create a two-tiered society where only the wealthy can afford to stay biologically young? Bioethicists at the Hastings Center have proposed a framework for evaluating anti-aging interventions that distinguishes between treating age-related diseases and intervening in the aging process itself. This distinction has significant implications for insurance coverage, regulatory approval, and public perception. Is aging a disease to be cured, or a natural process to be respected? Scientists in this field emphasize that the goal isn't necessarily extreme life extension but extending the healthspan, the period of life spent in good health. Dr. Laura Niedernhofer of the Institute on the Biology of Aging and Metabolism suggests that successful interventions might compress the period of age-related decline rather than simply extending lifespan. In this vision, people would live roughly the same number of years but would remain healthy for a larger proportion of those years, with a shorter period of decline at the end of life. The philosophical dimensions of anti-aging research extend into fundamental questions about human identity and the meaning of a life cycle. Leon Kass, former chairman of the President's Council on Bioethics, has argued that mortality gives shape and urgency to human life, that the limited nature of our existence is what gives it meaning and drives our accomplishments. In this view, extending lifespan dramatically might diminish the very qualities that make human life precious. On the other side of the debate, transhumanist philosophers like Nick Bostrom counter that aging is simply a medical condition awaiting treatment, no different in principle from other diseases we seek to cure. They argue that there is nothing inherently valuable about suffering and decline, and that extending healthy life allows for greater human flourishing and achievement. This philosophical tension between accepting natural limits and transcending them through technology runs through much of the debate around anti-aging interventions. It touches on deep questions about what it means to be human and whether our humanity is defined partly by our mortality or by our capacity to overcome natural constraints through innovation and discovery. Global perspectives on aging interventions vary significantly across different cultures and nations. In Japan, where the aging population presents immediate demographic challenges, substantial government support exists for longevity research. The Japanese Ministry of Health, Labor and Welfare has created accelerated approval pathways specifically for regenerative medicine, including anti-aging therapies. This reflects both cultural attitudes toward aging and practical concerns about supporting an increasingly elderly population. By contrast, European regulatory bodies have generally taken a more cautious approach, emphasizing safety concerns and social implications over rapid deployment. The European Medicines Agency has called for extensive long-term studies before approving interventions that modify fundamental biological processes like aging. Nations with younger populations, particularly in Africa and parts of Asia, have different priorities entirely, focusing more on infectious disease and maternal health than longevity research. These diverse approaches reflect not just regulatory differences but deeper cultural attitudes toward aging, death, and the proper goals of medicine. Some societies view the extension of lifespan as a natural evolution of healthcare, while others question whether resources might be better directed toward solving more immediate health challenges that affect younger populations. As CRISPR-based aging interventions move closer to clinical reality, the conversation is shifting from whether we can reverse aspects of aging to whether and how we should implement these technologies in society. The scientific progress in understanding the molecular mechanisms of aging has been matched by equally rapid advances in our ability to intervene in these processes using precision tools like CRISPR. This represents nothing less than a paradigm shift in medicine, moving from treating individual diseases to addressing their common underlying cause, the aging process itself. The first therapies targeting the aging process rather than individual age-related diseases will likely be approved within the coming decade. This medical paradigm shift challenges our conventional understanding of aging as inevitable and opens new possibilities for human health and longevity. However, it also demands thoughtful consideration of how these technologies should be developed, regulated, and distributed to ensure they benefit humanity broadly rather than exacerbating existing inequalities. The race to reverse cellular senescence represents not just a scientific frontier but a societal one, requiring us to balance innovation with caution, individual choice with collective impact, and immediate benefits with long-term consequences. Looking ahead, the future of aging research promises even more revolutionary developments. Beyond current CRISPR applications, scientists are exploring how artificial intelligence might accelerate the discovery of aging pathways and potential interventions. Machine learning algorithms are already being used to identify patterns in aging-related genetic data that would be impossible for human researchers to detect. Another frontier involves the microbiome, the community of microorganisms that live in and on our bodies. Emerging research suggests that the microbiome changes with age and may contribute to age-related inflammation. Crispr technologies might eventually be used to modify the microbiome to promote longevity. Quantum biology, the application of quantum mechanics to biological processes, represents yet another cutting edge. Some researchers theorize that quantum effects might influence DNA repair mechanisms and mitochondrial function, potentially opening entirely new avenues for intervention. As these technologies converge, we may witness a transformation in human aging that previous generations could only imagine. The question remains whether our social, ethical, and regulatory frameworks can evolve as quickly as the science. As with all transformative technologies, the ultimate value of CRISPR-based age reversal will depend not just on what it can do, but on how we choose to use it. We stand at the threshold of what may be the most significant medical revolution in human history. Through understanding and intervening in cellular senescence with CRISPR technology, we're not just treating diseases, we're challenging the very nature of aging itself. The science has progressed from observing the epigenetic clock to actively manipulating it, from identifying the molecular mechanisms of aging to reprogramming them. As CRISPR-based therapies move from laboratory to clinic, and as companies race to commercialize these breakthroughs, we face profound questions about how these technologies will reshape human experience. The ethical, philosophical, and global dimensions of this revolution are as complex as the science itself. Will we create a world where extended health span is universally available, or will these technologies deepen existing divides? Will longer lives lead to greater human flourishing or unforeseen social challenges? The answers depend not just on scientific advancement but on the wisdom with which we implement these powerful tools. What's certain is that our understanding of aging has fundamentally changed. No longer an immutable law of nature, aging now appears as a flexible biological process, one that we may learn to slow, halt, or even reverse. How we navigate this new frontier will define not just the future of medicine, but potentially the future of humanity itself. Thanks for watching. Please like, subscribe, and hit the bell icon for updates. Your feedback is appreciated. We hope to see you again.